Hello, this is the cell cycle and mitosis review video that corresponds to your test review packet that you should have. This video will give you the answers for your test review. Hopefully you've already tried filling it out some yourself so that you can just check your answers as you watch the video and listen to my explanations. So good luck, pay attention, and remember this is worth 5 bonus points tomorrow on your test. So tomorrow for your test, you're going to need to make sure that you understand and that you know the structure of a chromosome. So we know that the middle part right here is a centromere, and that's what holds the two sister chromatids together. And so this is a chromatid. Ooh, that's bad. And this is a chromatid. And we call them sister chromatids because they are identical copies of each other. Now, there is a phase of the cell cycle in which cells are going to stop dividing altogether. And examples of these are going to be your nerve cells. So these cells go into what we call the G0 stage. That's usually exiting from the G1 phase of the cell cycle. And they no longer divide, and they sort of just keep on living their life as normal and doing metabolic processes that are needed for it to exist, but they're no longer going through mitosis or cytokinesis. It's important that you know that cell division in bacteria is called binary fission. This is different from division of our cells. We call the division of our cells mitosis and cytokinesis. The phase cycle of the cell cycle in which DNA is copied, that would be the S phase, the synthesis phase. This is where DNA is replicated. Now, I know we didn't exactly talk about cyclins in class, but they are the proteins that control the cell cycle. They're called cyclins, and they determine when the cell gets to move on from G1 to S, from S to G2, and ultimately when the cell gets to divide. These are those internal factors that we talked about. You won't have any questions on your test about cyclins, but this is just something that I wanted to make sure that I exposed you to in case you do see this word on your STAR test in May. The phase of the cell cycle in which the nuclear membrane is present and DNA is spread out into chromatin is called interphase. Remember, interphase is made up of G1, S, and G2. All three of those make up interphase. Okay, type of cell division in eukaryotic cells that results in two identical daughter cells. That is mitosis followed by cytokinesis, which is the division of the cytoplasm. List the phases of mitosis in order starting with prophase. Remember PMAT, prophase, metaphase, anaphase, and telophase. This is where it stops. This is the end of mitosis. Cytokinesis is its own thing. So cytokinesis happens after mitosis ends. And remember, that's the division of the cytoplasm. And mitosis is the division of the nucleus. This is an easy question. Phase of mitosis that follows anaphase. You'll know that's telophase as long as you remember PMAT. There's lots of questions on your test about the order of mitosis. Make sure you know what happens. Phase of the cell cycle that follows G2. Well, if we look at our cell cycle diagram in your notes, you know that has to be mitosis, and mitosis starts with prophase. So specifically, prophase follows G2. All right, network of fibers that attach and pull the chromosomes apart is called the mitotic spindle fibers. All right, that's what attaches to the centromere of the chromosomes and pulls them apart during anaphase. Pictured right here, this is a picture of prophase. We know that because the nuclear envelope is starting to disappear, and we see our DNA is arranged and condensed in chromosome forms instead of in chromatin form. G1, S, and G2 make up interphase. Don't forget that. Area near the nucleus that contains the centrioles and helps organize spindle formation is the centrosome. This was mentioned in the packet that you completed in class. So the centrosome is the place where the centrioles are until they move around to the opposite sides of the cells during prophase. Right, the place in cell membrane of an animal cell that pinches in during cytokinesis is called the cleavage furrow. This only happens in animal cells, okay? So keep that in mind. In plant cells, they don't pinch to divide. Instead, they form a cell plate in the middle. So if this was our plant cell that was dividing, it's going to form a cell plate in the middle, which is going to turn into a cell wall. And that's how we get two separate plant cells, whereas in the animal cells, it forms that cleavage furrow like right here to divide the two cells in half. Okay, phase of the cell cycle picture here is anaphase. We know that because our chromosomes are being pulled to the opposite sides. Our sister chromatids are being separated by the spindle fibers. All right, part of interphase, in which the cell grows to mature size and carries out its job, that is G1 phase. This is the first phase of a cell cycle. The cell is growing, doing its metabolic processes. This is the longest part of it, of the cell cycle that it's going to spend its time in. Okay, in mitosis, this phase that follows prophase is metaphase. Again, refer back to PMAT, and you should be able to get it right every time. Phase of the cell cycle that follows S phase, that's G2. Remember, it goes G1 to S to G2, and after G2 is when we do mitosis or M phase. DNA that is all spread out in the interphase nucleus is called chromatin. Remember, that looks like a bowl of spaghetti. It's all mixed together. It's not organized, and you can't easily see that during interphase. The phase of mitosis, in which the nuclear membrane and nucleolus disappear and the DNA scrunches up into chromosomes, is prophase. So this is when our DNA is condensed. Okay, in this part of interphase, cells make the molecules and organelles needed for cell division. This is G2. This is the last fast growth that the cell is going to go through, and it's going to make the enzymes it needs for mitosis. All right, the two copies of each chromosome are called homologous chromosomes because they're the same size, same shape, and they carry genes for the same traits. Now, this is something that we are going to talk about more when we get to meiosis next week, and it will not be on your test tomorrow. All right, these structures of the poles, which attach to the spindle fibers and pull the chromosomes, are called centrioles. That's where our spindle fibers are originating from, so that they can attach to the centromere of the chromosome. The cell pictured above in this picture is in metaphase. We know that because they are lined up, the chromosomes are lined up down the middle of the cell. When chromatin scrunches together and condenses, we call it a chromosome. Chromosomes look like an X. One of two identical arms on that chromosome is called a chromatid. Specifically, they're identical, so we call them sister chromatids, right, because they are perfect matches. All right, phase of mitosis that follows metaphase is anaphase. Remember PMAT, and you should get that right every time. Phase of the cell cycle that follows G1 is S phase, where our DNA is copied. Remember, we go G1 to S to G2, then M. All right, the cell picture here is an anaphase because we can see the sets of chromosomes are being pulled apart to opposite sides of the cell. The phase of cell division, in which the nuclear membrane returns and chromosomes spread out to chromatin, this is the very last part of mitosis, or the butt end, and that is telophase, right? We start to see the two new nuclei forming in its own side of the cell. The cell pictured here is in telophase because we see the two new nuclei that are forming on opposite sides of the cell. We know that this cell has to be a plant cell because you can see the cell plate that is beginning to form right in between it, instead of a cleavage furrow, which we would observe in an animal cell. All right, any substance that causes cancer is called a carcinogen. Make sure you know that for your test tomorrow. There's a test question over that. Phase of the cell cycle after S phase, in which cells make the molecules and organelles needed for cell division. Again, that's G2 phase. Last little bit of fast growth before we divide the cell. Phase of mitosis, in which chromatids separate and move to opposite sides of the cell. That's anaphase. So in anaphase, our chromosomes are moving, being pulled by the spindle fibers to the opposite sides of the cell. The very first dividing phase, or phase of mitosis, is prophase. PMAT, never fails. Phase of the cell cycle, where cells spend most of their time. They grow bigger and they do their job as body cells. This is our G1 phase, the first step of the cell cycle. List the phases of mitosis in order, starting with interphase. So interphase is before mitosis starts, and that includes G1, S, and G2 phase. Prophase, metaphase, anaphase, that's mitosis. After telophase, mitosis ends, and we must do cytokinesis in order to divide the cytoplasm. The red arrows in the picture here are pointing to what we call the cleavage furrow, which is what happens when an animal cell must divide its cytoplasm during cytokinesis. We know that this is an animal cell because plants make cell plates, not cleavage furrows. All right, condition in which a cell grows too big for its DNA to handle the demand for information, that's DNA overload. We didn't talk about this, but this is what occurs, and this is why cells must divide. All right, phase of mitosis where the cytoplasm splits into two. Again, this is not a phase of mitosis. This is after mitosis, but we know the cytoplasm is dividing during cytokinesis. Name the phase of the cell cycle in order starting from G1. So we have G1, S, G2, M, followed by cytokinesis. All right, the last little bit of the review is just matching. Make sure you fill these out or try to fill them out before you continue on with the video. I'm not going to explain each one since I basically explained them all during the previous slides, so I will just hold it on here for about 10 seconds. Feel free to pause and go back if you need to re-watch something or hold on a slide longer than the time allows. All right, this concludes the review video. Hopefully this was helpful, and you should have all the blanks filled in on your review. Make sure you bring it tomorrow so that you can get your 5 bonus points on your test. Make sure you go back and rewatch any parts that you need to watch, as well as play the Schoology games, the links that are provided on the Schoology update in order to help prepare for your test tomorrow. So good luck, happy studying, and I will see you tomorrow.